welcome to Chestnut Chat for 2023. I'm not going to go through the whole spiel again, but I think that's all the housekeeping where we're at. I know the first question that you all have is, when is deregulation going to happen? What is the timeline? When can I get a tree? We will address that. It's not going to be, you will get a tree on September 17th, 2023. We're not going to be that specific, but you'll get some sense of a timeline. So between what Andy and Adriana and I and Lisa have to present, you'll get some sense of the timeline of how things are going and when things might be accessible to members and the public alike. So we do plan on addressing that. Let me make sure there's no great, my chat is showing disabled, anonymous attendee. I'm sorry about that. You should be able to do the chat, but you can at least get a hold of us in the Q&A. All right, so with that, I'm not going to take any questions on my housekeeping, but I'm going to transfer it over to Lisa to open things up and tell us all about the exciting things that have been going on and are going to happen within the American Chestnut Foundation. Lisa, can you take it from here?
Sure, thank you, Sarah. Can you hear me okay?
Sounds great.
Okay, I've had internet problems all morning, so I'm a little bit rattled by it. But anyway, welcome everyone. Gosh, it's great to be back on Chestnut Chat. It kind of feels like we did the first one in April 2020 when we were kind of, re-engaging with the community. But thank you for your patience as we needed to do so much hiring last year. We hired 7 new staff members and they're all going great guns. So there has been a lot going on, as Sarah said. This is our 40th anniversary year. That is really exciting. 40 years ago in June, we were incorporated, and look at where we've come, just a ragtag bunch of folks that had $600 between them to, very robust and on a national stage, a single species organization, which I'm so proud to be a part of. Because there's so much going on and we're still going to be doing some new hiring, we've initiated a new project management initiative called Monday.com. ESF is on it with us. So we're trying to keep track of all these things in a really transparent and organized way. So that's been kind of fun to do that. Andy and Adriana will talk about the D58, Darling 58 launch, but we do have a task force of really smart individuals that are pulling it together and thinking about how it's going to be when we hear the day of that it's deregulated, what's going to happen, how is it going to be launched. So that's happening. We're also updating our 2017 strategic plan. It's 5 years old now. Lots has happened between 2017 and now. So we are doing that, updating the 4 focus areas of science and technology, restoration, organizational advancement, and promotion and outreach. So, and we have leaders, volunteer leaders from our board helping manage that. We've got to grow to be prepared for what we predict is it going to be a tsunami of attention with the deregulation of Darling 58 and the documentary film, which we're finally going to get across the finish line this year. That started way back in 2016, but it had some fits and starts, and we're really excited about the process and the product that finally is going to come about. The official trailer launched on Giving Tuesday in November. So if you haven't seen the trailer, it's a 3-minute or so kind of snapshot of what the film's going to be like, and it's visually very beautiful. We're working on getting music from a well-known kind of bluegrass band, so that's got some things we got to tie up, but it's going to be launched on Earth Day, and we're going to have some private showings, so stay tuned for that, and that's going to hopefully garner a lot of attention. We also are working on our branding. We have different visuals throughout the organization, throughout the chapters. So we've hired a branding firm called Blue Ion. Some of you have actually spoken with some of the principals there, and we're working on new logo, potential new logos, and just really making sure that our branding and our visuals and everything is consistent, because consistent messaging is really important, and it's going to be even more important going forward. We're working on a new website, and that's taking a lot of time. That's really important. We need to be able to be ready to have the traffic that will be increased with these new initiatives like the launch of D58 and the documentary film. And we continue to have really strong collaborations with, of course, ESF. And we've renewed MOUs with a memorandum of understanding with the U.S. Forest Service and the NRCS. We just had a meeting this week with the Eastern Band of the Cherokee Indians. We went over that memorandum MOU. So none of these are stronger than our partners on this call at ESF. So I'm so glad they're here today. And last but not least, many of you know, if you don't, this is going to be my last year as president and CEO. It's very hard to leave a job you love, and I love this job, but I think new leadership every 7 to 10 years is a good thing, and they're going to take it to the next level. And I know the search is underway, and I'm not involved in it for obvious reasons, but I did nominate some folks from my conservation network who I thought might be good candidates. With my encouragement, I asked the search committee, which is basically the executive committee, if we could have a staff voice on it. And they agreed, and Sara is our staff voice. She's non-voting, but she does have a voice to be able to represent the staff from that perspective. So I'm really grateful to Sara for doing that. So I don't know the timing quite yet, but it's probably going to take a number of months to place someone. And I'm not retiring. I'm going to try to rewire and do something that I'm passionate about. I still have my health, and I have a lot of passion for causes, and I have a lot of energy. So I'm going to look elsewhere to see what next I can contribute. But I will be available for the new CEO when they're appointed. Phone call away. I intend to celebrate and support, but not be intrusive. So just be a resource for them as long as they need. So Brian Burhans did that for me. I was always really grateful for his graciousness, and I plan on doing that for the next CEO. With that, did I come under the right time frame, Sara?
You are way ahead. You're doing awesome. You've got us off to a great start, Lisa, which means I can ask you a couple of questions from the floor. And also, I want to let everybody know if, so we've got almost 250 people on here, which is awesome. You guys are great. I love, we've got so much reinvigorated interest in our Chestnut chat. But I want to let you know, we've got the public comment period link in the chat. We've got a link to the original petition that Andy threw in there. So definitely check that out if you have any questions about what all the research that's been done that went into the public comment period. That link is on there. And I think Jules or Anna put in a link to the trailer that Lisa mentioned. Robbie says, Lisa, the trailer is awesome and great job, and his novel draft is finally complete. Great job, Robbie. And then I had a question for you from someone. Oh, John Hempel asks, hey, John, in going through the APHIS comments, so both John and I have been going back and forth trading some of our top hits for the public comment period. John says, one theme that shows moderately often is the concept that D58 was federally funded. I think it would be a good idea to note somewhere for reference the level of federal funding that was actually achieved on this versus private and TACF, understanding that precise figures are probably difficult to come up with. Lisa, do you want to handle that, or do you want to pawn that off to Andy?
I think I'll pawn it off to Andy.
Okay. Well, let me see if there's anything else for you in here. Robbie says, we'll miss you, Lisa. You've done an unbelievable job, and we're so grateful. That's awesome. Okay. Well, I don't see any specific questions for you, Lisa, but if they come up, we'll bring you back in. Thank you so much for getting us off to a great and timely start. Andy, that means that if you need to go long, we might have the time, or we can answer more questions at the end. So if you want to take it away.
All right, thanks. To start with introducing myself, I am Dr. Andrew Newhouse. I go by Andy, and I work at SUNY-ESF, as has been mentioned. I am now directing the American Chestnut Research and Restoration Project at SUNY-ESF. I've recently taken over for Dr. Bill Powell, who has recently retired due to some health issues, but he is able to continue working part-time. And so he's still around and advising and helping us out. So we're missing his regular input, but appreciate that he's able to help as he can. So It's a big transition for me. I'm taking on some new responsibilities, but still primarily focused on the regulatory review process for the transgenic chestnut. That was the topic of my PhD dissertation, and really what's brought us to where we are now in terms of restoration potential. So I'll start with kind of an overview of that process, and then also fill in some details for where we are now, and where we are now, but also how soon we expect to get to the next steps. But also we have Adriana del Grosso, who is one of our colleagues here at ESF. She's our she's our distribution manager. She'll introduce herself more in a little bit, but she'll be filling in some of the details about at least preliminary plans for how we're hoping to be able to distribute transgenic material. So to start with the transgenic tree itself, I think I'll address the question a little bit of technical detail, not technical detail, get to the technical question, but a little bit of background about the tree itself before the regulatory review of the tree. So we have added a new gene to the American chestnut tree. The gene came from wheat, but it's not related to things like gluten or allergens. What the gene does, it makes an enzyme that breaks down oxalic acid, which is a toxin produced by the blight fungus. And when the tree can break down this toxin, that means it still gets blight, but the blight doesn't do as much damage. So it's more similar. It's not exactly the same mechanism as found in Chinese chestnut, but it's more similar to that scenario that Chinese chestnuts can get blight. They just don't usually die from blight. And that's what we've really accomplished with Darling 58. And so the gene or the new enzyme is called oxalate oxidase, and that has been thoroughly studied and reviewed and is currently being evaluated for distribution. The reason we can't just distribute it right now is because of the techniques we use. The genetic engineering process is reviewed by, is regulated, is covered by 3 different federal agencies in the US and other agencies in other countries as well. Even though genetic engineering makes smaller and more precise changes than things like traditional breeding and other efforts, the technology is tightly regulated where traditional breeding is not. So to get to the question from earlier about the impact of the transgene on the ecosystem and on degradation and so on, that is a very reasonable question and something we thought very carefully about. We wanted to make this tree tolerate or resist a fungal pathogen. And we also are well aware that there are a number of fungi in the same ecosystem that are really important. And we don't want to interrupt or disturb those relationships with beneficial fungi or other fungi in the environment. And so we've looked very closely at this, and we've done several studies. The link to the petition in the chat is probably the most comprehensive source for those at the moment, but we're continuing with even larger scale studies as we're able to moving forward. But basically, the mechanism of the transgene that's breaking down a toxin and not killing or repelling the fungus is really key here. And so it's not an antifungal gene. There aren't parts of this that are detrimental to the fungus itself. It's just breaking down this toxin, which prevents it from spreading as far and doing as much damage on the tree. So that was a really key theoretical concept for us, but in our testing and repeated tests, both in greenhouses and in field environments, we're not seeing differences in things like mycorrhizal fungi or other beneficial microbes compared to non-transgenic trees or backcrust trees or other types of trees. So we are fully confident that this oxalate oxidase enzyme that's present in Darling 58 is not interrupting relationships with other fungi. We've also done tests with degradation of chestnut leaves. Like decomposition is clearly this important process in a healthy forest. And so we actually looked at transgenic chestnut leaves next to non-transgenic chestnut leaves and watched how fast they degrade in different environments. And the short answer to that is that transgenic and non-transgenic leaves look the same. There are no measurable differences in the way those degrade or so on over time. We do see some differences compared to Chinese chestnut leaves. They seem to act more like oak leaves that are a little thicker, a little waxier. You can tell if you've held a Chinese chestnut leaf compared to an American chestnut leaf. They feel different. Yeah, they're going to act differently in the environment, and they do. But the transgenic American chestnut, non-transgenic American chestnut, backcross American chestnut, backcross trees that essentially look and act like American chestnuts are all in the same category of behaving about the same way in the tests that we've done. So with that, moving to the regulatory review processes. Like I said, there are 3 agencies in the US. The first one we'll talk about is the USDA. They currently maintain our permits. So we can only plant transgenic trees outside or move them between states if we have permits from the USDA. And these are not trivial. There are a lot of paperwork hassles and inspections. It's a very, very carefully controlled process. And so we do have the ability to test trees outside and do controlled pollinations on multiple sites. This has been really important to generate data, but it's not really practical for large-scale restoration plantings. And so the next step with the USDA is to file what's called a petition for non-regulated status. And the link, like I said, is in the chat. And that is mostly addressing, generally speaking, environmental risks. And specifically for the agency, they're interested in threats to agriculture and threats to other plants, and to some extent threats to human health. And so those are the biggest points in the petition. We are pleased that the agency has most recently published draft documents. That's what's open for public comment right now. But their draft decision documents say basically they agree that there aren't likely to be detrimental effects on the environment or on other plants or even on the chestnut tree itself, that this seems to be not risky, and therefore they intend to deregulate this. So they intend to grant us non-regulated status, which would mean we wouldn't have to worry about these permits from APHIS anymore, and we could start distributions. And that's what we'd like public comments on right now. So if you would like to weigh in on this process, show your support, even if it's something as simple as you like chestnut trees and you're excited about restoration, or you're excited about the potential for biotechnology to be involved in environmental conservation beyond just the agricultural applications that it's been used mostly in plants so far, please leave a comment. The links are in the chat, or in the chat at least. So moving on from the USDA, there are 2 other agencies that are involved. The next one I'll mention is the EPA. Environmental Protection Agency, in our case, regulates pesticides. Like I said, the oxalate oxidase is not a traditional pesticide in that it doesn't kill or repel the blight fungus. But their legal team decided that the oxalate oxidase mitigates the blight fungus. That's the key word in their law. And so they are regulating the transgenic American chestnut. More specifically, they're regulating the oxalate oxidase as sort of an active ingredient. They call it a PIP, or a plant incorporated protectant. And so they're focused mostly on that interaction between the oxalate oxidase and the fungus and potential environmental effects of that. And so we have turned in many documents to the EPA at this point. Their review is in some ways more complicated than the other agencies. And again, we're confident that they are proceeding with the review, even though this tree is totally unique compared to everything they've reviewed before. It's a forest type tree, not something that's confined in an orchard. And maybe the most unique aspect is that we want it to survive and spread in the wild. And that's exactly opposite of what most agricultural crops do and are designed to do. So we've turned in what's called a registration requesting a registration from the EPA, and also what's called the tolerance exemption. So that's basically saying there isn't a specific amount of oxalate oxidase over which it becomes harmful. Oxalate oxidase is already present in our diets, already found in the environment. So it wouldn't make sense to set a level, a dose rate of oxalate oxidase in this case. We've also turned in very recently, this is an exciting update, we've turned in what's called a petition for exemption from registration. And this will let us do large-scale restoration plantings. We don't know when. This exemption process doesn't have a specific timeline, but even while that's being reviewed, we are expecting that the registration, the first step in this sort of in our estimation, is that their registration will allow us to do plantings and distributions and allow us to get started with our existing goals and then over the next several years. So the third agency is the FDA, Food and Drug Administration, and they are much more specifically looking at health impacts, nutritional impacts for both human food and animal or livestock feed, and what the modification might mean for those nutritional or health impacts. And so we turned in extensive nutritional composition data, comparisons to things like known allergens. And the short answer there is that this doesn't resemble any known allergens, but they are also reviewing this. And so all three sets of documentation are in, all three agencies are reviewing them right now. And we expect to hear back this year from all three agencies for initial distributions to begin. That's so exciting to say because we've been saying for at least 10 years, we've been saying, oh, it'll probably take 3 to 5 more years, and just kind of push it off in that generic sometime in the future timeframe. And now we're expecting it's this year. So that's really encouraging. And what am I missing, Sarah? And how are we doing on time?
You're doing great. We're all ahead of schedule. This is amazing. So I have time for like the mini questions that are in here for you. Are you cool with where you're at? It sounds like you wrapped it all up. So can I will. Okay.
I will probably defer stuff about distribution plans until Adriana gets a chance to do her her introduction. But if there are questions on maybe the review process more specifically, what I've already covered, then sure, let's get to those.
Well, I like this question. I think I know the answer, but Chuck McQuaid, hi Chuck, asks, why was the comment period extended? And does that suggest that Darling 58 will not be approved?
We don't know exactly why it was extended. There were a couple of comments early on requesting that it be extended to just give more time for people to think about this, I guess, or to come up with their comments. I don't know. But it was only extended by 30 days, and that's still well within the estimated time frame that the agency proposed. So the last thing we've seen in writing from the USDA was in August of 2021, when they said they expected their final decision to be posted within 2 years. So that would put it at August of 2023. And so the fact that this is an extra month of time for people to submit comments shouldn't really change the final decision time. So what we have in writing is they expect it to be in August. We're reasonably optimistic it might be before then. So we hope that we might be able to do some spring plantings, potentially pollinations. That's not guaranteed, but we're hopeful, we're optimistic. And the same timeframes would basically apply to the other agencies. The EPA, we have verbal confirmation that they expect to be done with their review by June 15th, which is cutting it pretty closely for pollination season, I realize, especially in southern parts of the range. But even if we can do some pollinations this year on orchards that are outside of our existing permit structure, that would be really a benefit to enhance genetic diversity. I know that was one of the questions about a bottleneck. Let's come back to that later. I won't sidetrack it with it right now. But as far as the comment period, no, we don't really think that'll change the overall time frame.
Great. And let's go back to John's question that Lisa punt it over to you. How much federal funding has been given to this project and or could potentially in the future?
I don't know numbers, and I'm not even going to guess, but very broadly speaking, a majority of our funding, especially in the last 3 years, has come from private philanthropic foundations, specifically the Templeton World Charity Foundation. Wonderful. But other private donors. We've gotten significant funding from crowdfunding campaigns in the past, from just individuals who are excited about this project. And there are ways to donate to the college as a nonprofit that gets funding directly to our research project, and so that's been huge. We've also gotten significant federal funding. So I just yesterday finished applying for another round of funding on what's called the BRAG program from the USDA, separate from their regulatory departments, but it's still part of the same broader agency. And BRAG stands for Biotechnology Risk Assessment Grant. And so we all agree, yes, it's worth assessing potential risks of biotechnology before it's used for restoration. We've had 2 rounds of BRAG funding in the past. That's been pretty substantial. So federal funding for our research, and we were hoping to get more. And so the potential for planting trees for restoration will really be informative to us and to the agency, potentially to answer questions about future use of biotechnology. So that's exciting. But the funding has been a mix of federal and private philanthropic funding primarily.
Can I answer a little bit of that too as well? Thank you, Andy.
Please.
We have also done some co-fundraising together with ESF, and specifically the Foundation of the Carolinas has given a 6-figure gift every year for about 7 years. But as far as federal funding, we hope that increases. There's a lot of conservation and restoration language in both the infrastructure bill and the Inflation Reduction Act. We are gearing up a government relations team, and we are lucky enough to have Ambassador Kathy Novelli on our board, who used to be an undersecretary under the Obama administration. Also, we've got a new pro bono senior advisor in Holland and Knight in DC, that firm, who knows the Hill work and appropriations, affectionately known as appropes, and how we can maybe get in line for some of these and for a good fit. So Sara and I are planning on going to DC the end of February and perhaps having some meetings. We hope we can get increased federal funding, because it's going to take a civilian conservation corps level of programming to get this tree out in the forest. So we're excited about that. So thanks for the question.
All right, Andy, I got a couple of questions. So I think that whole 3 agency thing, like it often gets a little muddy as to what that is. So a couple of people want some clarification. Tom and Jerry, you guys are both together. I had to do it. They ask, do all 3 agencies have the option, if they decide to rule before late summer of 23? Can they rule early? That's question number 1.
Yes.
Is there a statute? Okay, so yes. Is there a statutory timeline? Are they being held to any sort of definitive timeline? Do they have to rule by a certain time?
Sort of. The EPA they were supposed to be done in December of 2022. They requested an extension, and that's what they said needs to be pushed to June for a number of reasons, but not because they're nervous, because this is different than anything else they've reviewed, and they have various staffing challenges and so on. So that got pushed to June. So yeah, the EPA is the only one with a statutory limit on their review timeline.
They can always request another extension, right? I mean, that's why
it's we don't really know. This hasn't been done before. There isn't a precedent for a wild tree to be released, but already their 6-month extension is longer than what we were advised might. Apparently, most of the time their extensions are 1 or 2 or 3 months, and for more familiar crops. So already it's longer than we expect. So I'm again hopeful that this will be it, but no, they have the power here to do what they want.
And last question about deregulation. And there probably will be more, but currently in here, do all 3 agencies have to provide clearance for D58 to become deregulated and distributed?
Sort of. Again, USDA, definitely. They control our permits. They currently say this is regulated. And once they give non-regulated status, the USDA is done and no longer regulated, no more strings. This is for Darling 58, at least. That one event, we can come back to that later, but USDA would be done and they have to give their approval of non-regulated status. EPA has to give us a registration, but there are different ways that might be implemented. There might be some sort of provisional registration. That's not exactly the term they use, but there might be some way they can say, this can be planted on a large scale, but not an indefinite forever scale while they're reviewing that exemption document we expect. So yes, EPA has to give us something, which is vague, but yes. FDA is unique because their process isn't technically required. They don't have to give us a yes, but since they're reviewing potential concerns with food safety and human health concerns, if they see anything that's concerning, they would get back to us and need to figure something out before this could be used as a food. That's also tricky because chestnuts are a food. People would collect these and eat them. They're delicious. We like that aspect of a wild tree that produces a cool, tasty, nutritious crop, and of course all the wildlife interactions that come with it. But we, at least in the short term, don't imagine selling transgenic chestnuts as a food product. So they wouldn't be bagged up as transgenic chestnuts in a grocery store to sell. They would be available to eventually forage as more of a wild crop or maybe integrate into breeding efforts, but that's down the road. Clearly transgenic chestnuts in the next few years are valuable for planting and we want to see these trees survive. But the question of selling them as a food crop is involved in the FDA review process as well, but not really a big issue, at least for the next few years. So hopefully that's sufficiently vague.
Right, which is why this makes it so much more complicated than it feels like it should be. Billy asks, so when oxalate oxidase breaks down oxalic acid, it creates carbon dioxide and hydrogen peroxide. Oh, I lost it. There we go. I've been told that the amounts of these are negligible, but I'm curious about the numbers, especially once D58 becomes widespread in the wild. Good
question. The numbers are very, very small compared to other sources of both of those things. Hydrogen peroxide, I'll start with that. I dove into this pretty deeply for the APHIS petition. So if you're really interested in the gory details, check that out. But hydrogen peroxide is not a stable molecule. That's why the peroxide you buy in the grocery store has an expiration date. It breaks down. And at the quantities produced in plants, it breaks down even faster. So it only exists as a molecule for a matter of minutes or maybe hours. And plants already make hydrogen peroxide as a signaling molecule. In some ways, or in some cases, it is made by a plant to stimulate existing disease responses. So there might be multiple benefits to having a little bit more of it for a short time in response to chestnut blight activity in the tree. But it's not building up to concentrations that suddenly make the tree antiseptic on any sort of scale. And carbon dioxide is a common breakdown product of organic molecules anyway. So the oxalic acid and various things that it's killing in the tree would probably end up as carbon dioxide eventually anyway. So, but again, the quantity of carbon dioxide that's released by this is minuscule compared to driving to the grocery store or something like that.
All right, there's so many good questions on here. I don't know if we'll be able to get to them all, so bear with me if we don't. I want to do this one. Don asks, if the agencies rule negatively, can that be appealed?
We don't know exactly how that'll work, but probably. And I think more likely, instead of just a ruling of no, would be getting back to us with questions for further information. So they might say, we can't make a decision until we understand what would happen in this scenario or something like that. And so we've already gone back and forth with the EPA on multiple rounds of questions like that. And so there could be more things like that where they just need more information in order to make a decision. But yes, if there is a no or a not yet answer, there should be ways to address that with further data or with refinements on the tree or something like that. But so far, every indication we've seen is that the agencies intend to do this, but the timing and the conditions will be tricky because there isn't a precedent.
Right. Well, there's a lot more. I have to pick one, right? That's a lot. I think I'm going to come back to some of these because I think I need to do some more curating of these questions. If you guys don't mind, I'm going to segue into my part. We're still 10 minutes ahead. We're doing great. Thank you all. This is going fantastic. We're not that rusty yet. Andy, fantastic, amazing information. You covered a lot of what people want to know. So I'm going to answer one of someone's questions. So Fred Behringer asks, there are over 28,000 comments which have been submitted. Do we have any idea in the percentage of favor of non-regulated status? So those 28,000 comments are for all 3 public comment periods. So there was one in August of 2020, there was one in like October of 2021, and there was one that then started this last November. This current public comment period, there have been about 7,300 comments, and currently it looks like the support is highest. What happened in August of 2020 is same thing. We had 60% supportive comments, and then there was a deluge of signatures coming in from those critical to deregulation right at the last minute. So we expect that to happen again. But I don't think that changes that generally there's a lot of favorable support out there on deregulation. Andy, I don't know if you have anything else to say on that particular question.
I imagine you'll hit it, but it matters to the agency whether comments are unique. So if they get 1,000 form letters that are identical, this is oversimplified, but they can sort of treat that like one thing that they have to respond to and not 1,000 things they have to respond to. So unique comments matter. And like Sara said, the many signatures opposed would not necessarily be considered the same way as unique comments.
Great. Well, so I'm going to talk a little bit about, I know you guys all want to know about distribution. Adriana is going to cover some of those specifics, but I want to talk a little bit about what we want to do within the American Chestnut Foundation and through ESF to make sure you guys are all ready to do the few things that we want you to do to be ready. And I'm using my cheat sheet. This is the agenda that I drew up, and I'm using my phone here. So if you see me looking over here, I'm working. So plans for diversification and ramping up. So one of you asked, are you worried about genetic bottlenecks? Yes. And so I want to give many props. The reason Jared is on here as a panelist is because I may get in over my head here. Jared is doing a bunch of this amazing work with some colleagues at Hudson Alpha and Virginia Tech to make sure that we don't hit those types of genetic bottlenecks. So through the American Chestnut Foundation's efforts, we have many orchards throughout the Eastern US. We have over 500 of them. There are some that we call GCO, germplasm conservation orchards. Those are wild American sources. We also have many backcross orchards. And I saw a couple questions in here about the status of the backcross breeding program. So Jared, can I turn this over to you? Because I think you have these talking points a little bit better, concisely put together. Could you do about 5 minutes about the current status of the backcross breeding program, how we're going to use them for diversification, as well as how we're going to use wild Americans?
Yes, so hi everyone, Jared, Director of Science for the American Chestnut Foundation. So one of the things we've done with the backcross program is we have crossed people over many years, like 40 years now, have been finding wild trees and then taking pollen from our hybrids, putting on those wild trees, harvesting the nuts, and then planting them in orchards and inoculating them. And so these trees are, 15 years old. Many of them are flowering. We have worked with Virginia Tech to genotype the trees to figure out how much of their genome was inherited from Chinese chestnut versus American chestnut. And so a lot of these trees, in fact, some of them we've done back crossing so well that they're pure American, and they're flowering in orchards. So we're prioritizing trees for Darling 58 pollinations that have the healthiest trees with the most American genome, basically. And the goal when we do these crosses is with Darling 58 pollen, which is going to be basically 100% American plus this wheat gene, is to select progeny from those crosses that are pure American. So, we're going to get rid of the Chinese genome component. There was a question I saw around, like, do you want to combine oxalate oxidase with these different mechanisms of resistance in Chinese chestnut? And we did an experiment. We're continuing to do it this year, but we did an experiment last year where we bred Darling 58 trees with some of our better backcross trees that have resistance to blight, partial resistance. And we wanted to see, is there an increase by combining the oxalate oxidase with many genes from Chinese chestnut that are contributing to resistance? And we actually didn't see a strong effect with the Chinese chestnut genes. We saw that the oxalate oxidase gene seems to boost resistance similar to Chinese chestnut, and that there wasn't a really strong effect of these other genes from Chinese chestnut. But we still want to plant the trees out over the long term and see how they, fare with the blight over many years. So we're doing those kind of crosses of like seeing if we can combine and stack resistance from Chinese chestnut plus this oxalate oxidase gene as a research question. But in terms of our initial distribution, our goal is to diversify the population with backcross trees and trees that wild American trees that we've conserved in orchards that represent 99% of the adaptive diversity in the species. And we figured out from sequencing 400 American chestnuts from across the range that we can capture 99% of the diversity by breeding with 200 wild type trees from across the range. And we have 400 or so of these backcross lines and trees conserved in wild American trees conserved in orchards. So we have more than enough to represent the adaptive diversity in the species with the material that we have that's available in orchards. So we want to use the backcross trees for the diversification, simply for the practical desire to have these flowering trees that are available in orchards. And our goal is really to select out that Chinese genome because we don't see it's really contributing to the resistance. So yeah, that's the main things we're doing.
Thanks, Jared. And I want to make a couple points. So Jared, the amount of analysis and work that has gone into that little 5-minute blurb is just incredible. So I want to thank you and all of the chapter members. Many of you on here have collected leaf tissue that have gone into these analyses. How many trees have you genotyped as part of this work to go into this?
I think we're up at 5,500 so far, at least in the backcross program. And then we started genotyping the transgenic trees. And one of the things I didn't mention in my previous blurb is we are worried about the bottlenecks. So we're starting the population from the Darling 58 tree, which is a tree from New York where the wheat gene was inserted. And it's called the wild tree we call Ellis from New York. And we know the whole genome of Ellis. We sequenced it, and we can see when we do the crossing with Darling 58 to all these wild trees, we can see how much of the Ellis genome is present with the DNA markers. And we can select against the Ellis genome and select for whatever tree's genome we're crossing with. So a tree from Georgia, we can select for more of those genes from Georgia trees. Tree from Maine, we can select for more of the genes from Maine trees. So what we're trying to do with the genotyping, we're going to genotype all of the material that comes out of the crosses that we're making with Darling 58. And we're going to A, select out Chinese genes, and B, select out Ellis, and make the trees have more diversity from the wild population and reduce that bottleneck. So that we have like a pipeline to be able to actually do that, to see the Ellis and get rid of it.
And I think one more point to make too about the backcross populations. I saw a question in there that says, you know, there's a backcross planting nearby. Should they cut it down? And absolutely not. I think there are a few reasons for that. Number one is what Jared covered with the diversification, but also some of our backcross orchards have Phytophthora resistance. That's going to be essential for dissemination of populations in areas where Phytophthora cinnamomum is prevalent. And so there's a lot of other reasons that those backcross orchards are being utilized. We also have an extensive program of pyramiding resistance within the backcross population for another traditional breeding arm. While the study, Jared, that you mentioned said that there's not a lot of measurable resistance, may I caution that that's one study, that there are others ongoing, and that we have seen some measurable resistance in those populations, and we're continuing to investigate that. Is that a fair statement?
I mean, we're also doing, when in characterizing our backcross trees, we've been looking at we've been like assessing their resistance, and resistance is a spectrum. And we see that the backcross trees, we're trying to select trees that have resistance similar to a 50-50 Chinese American, but they have, let's say 80% American chestnut genome, but they have resistance similar to a 50-50 hybrid. So we are selecting trees in our backcross program that have above average resistance, and then we're crossing them or breeding them together, and we're seeing if we can select their kids that have resistance even better than their parents. So that, you know, we're continuing to do that work and to see how effective that is. What we have seen with the Darling, you know, comparing backcross trees to Darling, is that the Darling seems to have very high levels of resistance, and that's why we're all in on this. Like, we really, it's the most effective path right now that we know of for enhancing blight resistance. That doesn't mean it's the end, you know, like the final product. There might be ways that we can, you know, express the gene only when the tree is wounded, or figure out different genes that we might stack, you know, using biotech with oxalate oxidase. You know, we're still open to continuing to improve on what we're doing, but right now, the most hopeful approach for blight resistance is this Darling 58. So we're all in. We're all in.
Absolutely. All right. So Brian asked, and Jared, you and I knew this question was going to come up. Brian says, don't we already have 200 plus diverse wild trees? Do we really need backcross trees?
I don't think we have 200. We might have 200 wild trees planted in orchards, but they might be derived from like one wild tree. So like you can, you know, we have a lot of, that's a kind of like a finer point, but let's say we've been collecting seed from these wild trees and planting them in orchards. And it's like one tree, we plant, you know, 10, 20, 30 seeds from that one tree. So we've only represented the diversity from one tree, even though we have 30 of its kids. And when in our germplasm conservation orchards, I don't really know how many total we have, but it's less than what we have in the backcross. I'm still looking into like, what do we conserved in the germplasm orchards in total? And it's probably in the order of like 50 trees. I don't know, Sarah, what you think we've actually conserved.
That are flowering? Probably less than 50, and I think I also want to address, I think I see this a ton on Facebook and on the various listservs, this idea of purity and what it means to be an American chestnut. And so I feel this, and I'm reading into your question, Brian. I'm reading a lot of sort of knee-jerk, like, it's not pure, you know, we need 100% American. And I think when we say backcross trees, we also mean some of them have no Chinese background in them whatsoever. They're in a backcross orchard. They went through the process, but for whatever reason, they are 100% American chestnut. They just weren't planted as that. And like Jared said, to his point, we can also select out those portions of Chinese genomes to get a fully wild type. So I think that a lot of that concern is we can eliminate a lot of that concern. Just because a tree is backcrossed doesn't mean that it's not very close to or 100% quote unquote wild type. And Christopher asks, no, it wasn't Christopher, it was somebody else. Oh, John says, by genotyping, does that imply full genome sequence or something less detailed?
What we're doing to make it, we have done full genome sequencing, but when it comes to this method of selecting out the Chinese in the kids of the Darling 58, when we cross with backcross trees, we're using we're using 5,000 markers distributed across the genome. So like we are able to see and these particular variants in the genome are fixed for a certain, variant of a gene in Chinese and fixed for a different variant in American. So we can tell this is Chinese, that came from American. So we're able to see with very high precision that this tree that came out from the Darling 58, where maybe the mom had 3% Chinese, we can select out that 3% in the kids with high precision using these genotype markers.
Jared, I'm so happy you were on here to answer those much more concisely and thoroughly than I could. Thank you. I only have a couple more minutes, so I see a lot of questions on there about pollen distribution, how we're going to be able to do that. There's two things that you need to have. So when deregulation happens, maybe it'll be in time for this pollination season, maybe it won't. But two things you need to have in order for us, the collective us, the royal us, to send out pollen. Your tree needs to be logged in Dentatabase. Have you guys heard me talk about Dentatabase? If you haven't, then right, Lisa, it's not a program with me unless I talk about Dentatabase. So that's going to be one requirement. We do intend on having one or more trainings on Dentatabase. I've had I love the laughing face. That makes me very happy. We're going to have one or more trainings on how to use the system. We're feverishly making some improvements to the system to make sure that you guys can easily get your trees in there. So if you have wild trees that are in Dentatabase that have already been entered, you'll be able to use that and pollinate them in the system, and then follow the harvest data and information. That's going to be really critical for us being able to track this. I saw another question in there about how much are you going to track it? Well, that's the system that we'll be able to use to see who gets what pollen, what trees are those pollen types going on to, how much diversification have we been able to achieve. It's all going to be done as long as we have some sort of really structured, not 100% structured, but really structured program of tracking all of this. And we're going to be doing that in Dentatabase. So if you don't have an account, we will do our best to keep up with the requests to make sure you have an account and also get you in the system. Like I said, February, March, April, we're going to have one or more trainings on how to use the system. If you were in the system a few years ago, it is a lot better now. So don't just say, I tried it a few years ago and I didn't like it and it was hard to use. I know it was, but it's a lot better now. Many, many thanks to Lisa's fundraising and efforts and support toward that program. Kendra is also going to take a few minutes at the end of this program to talk more about how that functions and what it looks like. So stay tuned for that. Pollen distribution. So you need to have your trees in Dentatabase. You also need to be trained. So we're going to have a series of trainings of how to many of you are already trained on how to pollinate. We're going to have at several strategic locations, we're going to have kits available, pollination bags, ties. We'll do some trainings this summer. So if dereg happens this summer, we might be able to do it. Can I call it dereg? Is that lame? Andy, give me a nod that I can continue using
close
that
enough.
term. Okay, close enough. So if we can happen this summer, it will. If not, you'll be trained for next year. Do you have something? Unmuted. So
I was going to jump in, not to change plans, but to add the regulatory, of course, strings that might be attached. That especially from EPA, it's not entirely clear exactly when we'll be able to, or how we'll be able to distribute pollen. This is, again, something new for them. We don't have a precedent. We absolutely intend to continue working with TACF as a collective group to pollinate diverse American chestnut and appropriate backcross trees in as many orchards and locations as we can to get those genetic diversity benefits and to advance the restoration efforts. That's the plan. But since this is new, especially to EPA, there might be limitations on how many places we can do pollinations, especially the first couple of years. So yes, you will be able to do pollinations eventually, but I don't want to promise that, can't promise that it will be sent to every backyard, especially the first couple of years. It might be limited to a few TACF orchards or something like that. We really don't know what the conditions will be, but expect it will be trickier for pollen than it will be for seedlings. So hopefully that's
I think that Andy, I appreciate you mentioning that. And I think it's not only a regulatory aspect, which is really important. I mean, that's number one, right? We want to make sure we're following the rules. But number two is just in terms of resources and how quickly can we do diversification, it's going to be a lot easier for us to pull a bucket truck into a location that has, so this is why we set up the GCOs the way we did, or why we're using some of these backcross orchards and really picking out the really best individuals to use for this crossing is, we can maximize our diversification efforts in a location that has lots of trees and people are trained. So that's not to downplay those of you who have a few trees in your backyard or one or two. You will have an opportunity to do that, but it may not be of highest priority in the first year or two. Is that a fair way of restating that, Andy?
Sure.
Okay. We're working with lots of nurseries to make sure that we have a pipeline. Those of you who have already ordered seedlings from the American Chestnut Foundation have seen some of that. We're working to improve that that pipeline and distribution model. So Stephen Hoy, who works with me here at Penn State, is doing a lot of amazing work. So is Sierra Wilbur. She's our nursery manager at Meadowview. She's doing amazing work with that as well. We're looking at containerized production methods as well, and nursery partners. So I see a lot of questions in here. How am I going to get nuts or seedlings? Adriana's going to cover some of that here in just a minute. So we're going to get to it. Again, it's going to be a really small supply in the first few years. And it's going to be, it's not going to be full open. Everybody gets a seedling, you know, right as soon as deregulation happens. There's still going to be a time for ramping up. So John says, what's the fastest path to widespread propagation of seedlings and nuts? Is it pollination to non-resistant trees, tissue culture, et cetera? Definitely not tissue culture, at least not now. Tissue culture in chestnut is difficult, costly. So it really is going to be doing controlled pollinations for the diversification and then open pollination for a lot of seed production. That's really the model that I think is going to be most appropriate for getting lots and lots of trees. The last thing I want to cover before pushing this over to Adriana is the educational resources. So these chestnut chats are all in an archive. They're all on YouTube. We have a few that are on site selection, and we have 2 that are on pollination. So go back and harvest and things like that. Definitely go back and review those. We have tons of fact sheets on our website. Like I said, we've got plans on doing some planting webinars. We will definitely get those chestnut chats either by this fall or next spring, so that you all are prepared for site selection. That's number one. You got to make sure you have a good site that's appropriate for planting these things, and that you can start doing the site prep work and everything that needs to be done prior to planting, so that you're ready for that. I think I covered all of that. You guys feel like that session's good and we can move on to Adriana? Okay, we've got 28 minutes. But to do Adriana with distribution and Kendra with dentatabase. All right, Adriana, you're next.
Hi, my name is Adriana Del Grosso. I'm relatively new to the ESF project. I just started about a year ago. I am the tree distribution manager for the project. And this has been a really great year of just learning about this really fantastic network of American chestnut enthusiasts, and learning about all the amazing resources and history of the American Chestnut Foundation and all the great work they do with volunteers in the conservation world. And yeah, I'm excited to be here, work with all of you. Yeah, so my position is primarily outreach and education and laying the groundwork for the initial stages of distribution of our trees, which, as you all know, through what Andy has told us, we cannot distribute our trees just yet. So yeah, just laying the groundwork now. So what that looks like is a lot of coordination with the American Chestnut Foundation, because our plans are their distribution plans as well. We don't necessarily have our own like individual independent distribution plans from ESF. We're really going to be tapping into the vast network of the American Chestnut Foundation for our distribution plans. So I help with building partnerships for the first stages of distribution, work with establishing small scale plantings for educational purposes, eventually larger plantings. So I speak with some land managing organizations, some nonprofits, but yeah, very limited distribution right now, and focusing mostly on that education and outreach aspect and communicating our science to our collaborators and kind of working as a liaison between our project and TACF. Yeah, so I can tell you a little bit about our plans for distribution the first couple of years, and I would like to emphasize again that we won't have a lot of plant material, Darling 58 plant material available the first year or two, especially after deregulation. Deregulation doesn't necessarily like open up the gates to have trees in every household. That would be great. We want to make sure that everyone who appreciates this tree can get one, but that won't be realistic right off the bat necessarily. What deregulation does mean is that it will allow us to more openly pursue our goals for research and production of these trees. So open up possibilities for further outcrossing and for doing further plantings to study how these trees interact with the environment, study how we can better our goals for conservation and restoration of these trees. Yeah, so basically the first year, especially into the second and third year, our four main buckets, you could say, for distribution will be distribution for research purposes, for production purposes, for educational purposes, and then distribution to the public. Yes, and again, want to make sure that we can meet our goals for studying these trees, for outcrossing them, and for increasing production. And some things that might initially stand in the way of distribution to the public that we can hope to alleviate the first couple of years as we figure out how to work within this regulatory framework and how to build like an efficient strategy for distribution. Some of those bottlenecks might be making an efficient way to test Darling 58 nuts to make sure that they have inherited the gene for blight tolerance, because at the moment we have to do that manually in our lab. So once we make this process more efficient, we'll be able to test more and more nuts and seeds and make sure that they do in fact have blight tolerance. And in fact, in the future, it might be the case that all of these nuts have the trait, but for the time being, we have a kind of inefficient process for testing all of these nuts. Also, in terms of just production of seedlings, once we have those nuts that we know have tested positive to have the gene, we have fairly limited capacity to grow them out here at ESF. We are a research facility, so again, tapping into that network of TACF and also working with commercial producers, commercial nurseries to grow those trees out for distribution to members of the public who want to plant trees at home, and also for more large-scale restoration plantings so we can get these trees back out into the forest. But it will take some time to make those connections with nurseries and have them to the point where they are producing the thousands of trees necessary to introduce the trees back out into the forests and to get them out to the public. Yeah. But yeah, we hope that this will expand within the next couple of years to make more broad distribution possible. So briefly, to answer the question, like, when can I get a tree? Probably not within the first year, hopefully within several years. We are not intentionally slowing this process. It's going to be kind of like, it'll be a little while before we have an efficient system for getting these trees out. It will take some time to scale up what we are capable of doing and get from small production that we have now just to meet our small goals for outcrossing our trees and for research to get to the point where these trees are commercially available. But we'll get there. Thank you for your patience. We hope that distribution will be somewhat, or I guess, meeting our goals that are tied to distribution. Our goals for research will be, and education will be, a kind of interactive process. So some things that we are doing to involve the public and members of TACF are, we are establishing a small number of educational plantings of the transgenic trees. They will be some of the first trees planted. Part of my job is working with organizations to establish these small educational plantings in public spaces where they will be incorporated into educational programming so that more people can learn about the American chestnut and about biotechnology for conservation, to get the word out there so we can get more supporters. We'll also, as Sarah mentioned, be doing small scale pollen distribution that we hope can involve as many people as possible, working within the framework that the EPA might set as to how we are able to distribute pollen. So definitely trying to get more people trained on how to do these controlled pollinations with the transgenic pollen, which might involve inviting members of TACF to orchards to help us with pollinations, to again help us with our goals to outcross our trees and increase genetic diversity, capture the surviving genetic diversity, and also to get more people trained on how to do these pollinations. So if we are able to distribute pollen more broadly so that people can do pollinations on trees in their backyard or trees that they know of, then that would be great, just to tap even further into this broad network of enthusiastic volunteers who are willing to put in work. Yeah. And again, just to plug Dentatabase, we also hope that this distribution will be interactive in helping us further our goals for research, because people can track their plantings in Dentatabase. It's a great resource for us to see what exactly is going on, to see where the trees are going, how they're doing, so that we can better figure out how we're doing in terms of restoring these trees
So Adriana, can I use that as an opportunity? I want to do 2 questions and then segue to Kendra so that we can, is that okay if I
Yeah, absolutely.
Okay. I thought I heard Dentata base, so I know like that's me, that's my sign to come on. So I have one question in here that says, and I can't remember who it was, and I'm losing it because there's so many questions in here, but they say, I got some Dunstans. Can I plant these next to them? Are some of these restrictions going to be that I can't plant them wherever I want? What do you think about
that? As long as you're okay with having nuts that are a hybrid between the American chestnut and a Dunstan tree, that's fine.
So I think the question is, there's no regulatory reason why you can't plant these near other chestnuts. I know there is concern from the organic growing community. So I would say that if you are neighboring an organic chestnut producer, you might want to keep your trees away from there just to stay good neighbors, because while the organic standards don't require that you do that, and the organic standards do allow some influx of genetically modified pollen onto organic seeds, there is some concern about certification within the organic community. So I say that's the only situation I could think of where you can literally plant them anywhere, but it might be, up the neighborly way to ensure. Andy, you might say this more eloquently.
Add 2 things to that. One, when we think about pollination and planting next to something else and potentially pollinating it, we're talking about like 100 yards. Chestnut pollen can go a ways, but it doesn't stay viable in the air or on insects for all that long. And so as long as you're not planting trees like just over the fence from an organically certified chestnut producing orchard, which there aren't very many in the native range of the American chestnut tree, I really don't think this will be a common issue. So realistic pollination range of maybe 100 yards under normal conditions is important to keep in mind. But yeah, to clarify, plant the trees where you want. Once you have a tree, we're not going to tell you where to plant it. I would also add to that though, if you are interested in participating with our broader goals of contributing back to the restoration efforts and potentially crossing trees, we wouldn't necessarily want Dunstan hybrids incorporated in forest restoration settings, because Dunstans aren't very well adapted to that. But anything that looks like an American chestnut and seems like it will be interacting well and adapted to Eastern US forests could absolutely be. Or if you want to cross these with Dunstan chestnuts to raise more trees to attract deer, okay. We're not specifying that, but we would distinguish what's incorporated back into the program for restoration purposes or research purposes, as opposed to what's done privately for your separate intentions.
Thanks, Andy. And then one more question, Adriana, that is for you. And I think, so Tree asked this way back at the beginning, almost an hour ago. Can a Master Gardener Association that manages a native plant arboretum in a county park buy seed or create an MOU or Board of Supervisors? It sounds like something like that might fit under the educational program. What are your thoughts there?
Yeah, that would definitely fit under the educational program. Right now, I think we are about at capacity for that program in terms of like the initial number of plantings we can handle, because we really want to focus on having like quality associations with these organizations rather than just getting trees out everywhere. Yeah, trees will be very limited. So unfortunately, I can't guarantee every potential educational planting site a set of trees, as much as we would love to just get them out there so that more people can learn about it. Yeah, can't guarantee Darling 58 trees for any educational site this first year. But yeah, you can always send me an email. My contact information is on the esf.edu/chestnut website. If you're interested, could potentially put you on a list for future distribution, or send you some educational materials. so that you can at least do education and outreach about chestnuts and then maybe plant some non-transgenic trees in the meantime.
I think that's a great point, Adriana. You know, planting non-transgenic trees, getting ready for the, I'm going to plug joining the American Chestnut Foundation. I think that's always going to be the best way to get the most up-to-date information and the most, we are working very hard collectively to increase production and so that we can have material to distribute for, like Adriana said, research purposes, educational purposes, and then member and collaborator and supporter distribution. With that, I'm going to move on to Dentatabase. Kendra, you are up. Or how's things going with your helper there and your internet?
I think my internet's fine. My helper is downstairs eating lunch, I hope. I've just been furiously trying to answer all the questions in the Q&A, so I apologize if anything I say has kind of already been covered. I'm Kendra. I'm the science coordinator for New England. I've been Sarah's partner in crime, developing the database for the past long number of years, over
12
10
years.
Yeah, since 2009 I've been involved with it, so it's been a while. Anyway, so bit of a spoiler alert, but a lot of what I'm going to cover is also going to be outlined in the RSC column in the upcoming Chestnut magazine. So for those of you who are members, you don't have to take furious notes. It's all going to be written down for you, and there'll even be a few pictures. But, you know, as we've kind of talked about this morning, or I guess it's this afternoon now, distribution this is a big thing for us to get our arms around, and we're trying really hard to develop systems or use systems to keep track of what the heck we're doing. Everyone wants this pollen, everyone wants these trees, everyone wants to pollinate their trees. The excitement and enthusiasm is fantastic, but it also leads us to the very real possibility that we can quickly get really out of control and not know what's going on. So our database, thankfully, is a system we've been developing for the past over 10 years. It's designed to track all of our scientific data. So it tracks wild trees. I saw some questions in there about what kinds of stuff it tracks. It tracks wild trees. It tracks harvests and cross data. It tracks the plantings that those harvested nuts then go into. So there's a lot of, it tracks distributions. So when we send you material, depending on who you get it from, some of us are better than others about remembering to do that, but we can record those distributions. You know, I've seen some folks on here, I've sent seeds or scion material even to you, like those are in there. And so we want to make sure that we have the ability to track what we're doing with Dentatabase in terms of our pollinations this coming year. You know, fingers crossed, that's what we're doing. And so we're trying to be really ready for that. And so that means if you have wild trees that you want to pollinate, we need to make sure that they're tracked in Dentatabase. If you've been tracking them in TreeSnap, that's great, but that doesn't get them into Dentatabase. So we do have a tree locator program where you can send in a tree locator form. You can note the TreeSnap ID on that form and send us a sample. One of us on our science staff, you know, whichever science coordinator is closest, send, or I guess whichever is the appropriate science coordinator. Sometimes it's not the closest. I get a lot of samples for New York because I'm in Vermont, but Sara is actually the contact for New York. But we can assess those and enter them in Dentatabase. So if you've got a tree that you're thinking about, either ask us for an account, we can show you how to use it, you can look them up, or even contact, you know, your science coordinator, we can check in the system and see if those trees are in there. More likely, a lot of our pollinations this first year are going to be in orchard settings because we really want the biggest bang for our buck. As we've already talked about this morning, some of our backcross lines represent a lot of genetic diversity, and it's concentrated in one place in an orchard that is presumably a little easier to access than traipsing around in the woods. And so those orchards should be tracked in Dentatabase, but if they aren't, or if there's issues with that, we have some time to correct those and make sure those are in there. You know, I think those are kind of the big things. So, you know, once we are distributing pollen, we do want to record those crosses in Dentatabase. It's pretty straightforward to do once you know how to use this system. And that's, again, any of this data tracking is stuff that your science coordinator can help with. We'd love more people to know how to do it properly so that we can keep up with it. I always have a stack of things that need to get into Dentatabase that I haven't gotten to yet. So it keeps us busy, but as Sarah mentioned, we're going to do some trainings. I know I've done a lot of individual trainings with folks to teach people how to enter wild trees, how to enter observation data on their plantings. And we can organize those for your chapter. If your chapter is interested, we can do those regionally. I know Sarah's been threatening that we're going to do a big Dentatabase training, maybe not as an official Chestnut chat, but something similar sometime soon. So keep an eye out for those opportunities, or certainly reach out to us if you want to learn how to use the Dentatabase. We have a testing server that we often set folks up on first. It's where we push any new improvements to the system or any fixes, but it's also a really great learning environment because you can bang around in there. You can delete stuff, you can add stuff. If you break anything, it's no big deal. It's not our live database. And so that's been kind of a nice side benefit of that environment, is it's a great place to invite people into to learn how to use it. And learning by doing is really the best way to get ingrained. I think the last piece that I should just mention is we do have a new planting module that we're working to really improve by the time folks are getting D58 seedlings, to make it really easy to point and click on a map and plant your tree so that you can track those plantings. The first thing we're going to be distributing is probably going to be pollen. So that's been a bigger focus. But that planting module is going to be available for getting those early plantings in, especially because they will be small numbers of trees. And for those of you who have backyard plantings with just a couple of trees, if they're not in our system yet, that would be a great way to get those entered. So lots of things we can show you how to do. I have 6 minutes, so I'm probably not going to teach you how to do any of it right now. But again, reach out. Sarah and I, as well as Jamie and Brianna, are all the science coordinators for the organization. We're all happy to set you up with training and testing accounts and whatever you need. Anything I missed?
Well, Brian asks, does the new Dentatabase have a user manual? Last time I looked, it was the old version.
We just haven't updated the user manual. It's a huge static document, and the database is ever evolving and changing and getting improved. And so we've kind of moved away from having a static user guide document. We are hoping to incorporate more context sensitive help in the database, so that as you use it, there are ways to get support or get your questions answered. But no, that is woefully out of date, and I don't really see us putting a ton of effort into updating that anytime soon.
But I will say that despite its outdatedness, there are some excellent terminology and usability things in there, and that user manual, the stuff in there is more geared to what we're going to call a power user. So especially for those of you who have big plantings, and I'm talking like 50 to more. I mean, there are several of you that I know have 1,000 or more trees. That's the version you're going to want to use. For those of you who have 3, 5, 10, maybe 20, this new module, you don't even need a user manual for. Like, it's going to be so easy. Like, you go in, you point, you name it, and it's done. So the user manual, while slightly outdated and does not cover the new module, it is still valid and covers a lot of the stuff that's hard to do in Dentatabase, especially if you have lots and lots of trees. But if there are few, it's going to be super easy. It's going to be point and click. You're not going to need a reference. Is that fair, yeah. Kendra?
I think that's fair. I mean, I think the biggest thing, thing I would note about the user manuals, I think there's a lot of cautions in it about breaking things or entering things because we didn't have good controls. And so everything was a text field. And so it was really easy to not follow, kind of set nomenclature or set process that, we've been using for data entry. But moving forward, I think it makes it sound a little easier to break than it is now.
And I just saw a note,
which is thankfully a good thing.
Yeah, and Asunta's on here. She has been she
Asunta.
and Jack Ostroff. I don't know if Jack's on here or not, but I want to give huge, amazing props to them, our developing partners in the Tidbit out in California. Fern Coffey, who is a graduate of ESF, he's been helping us with Dentatabase. So this is a huge effort that lots of people are taking part in. There are so many questions in there. I see Jared like feverishly typing, Andy, Kendra, Adriana, you guys we're trying to answer all these questions, but we are pretty much out of time. I want to thank you guys we had almost at one point, we had almost 300 people come today. This was a lot of fun to give you guys an update. We don't yet have a topic for March. Tentatively, we are going to try and get the students who won our poster contest at the annual meeting last year. We're going to try and get them to present their work. That's the tentative topic for March. That'll be, I think, March 17th is the third Friday in March. But we're skipping February. But other than that, keep an eye out for some Dentatabase trainings and all the really exciting things that are coming out. Lisa or Andy, as the respective directors of your institutions, do you have any final words you'd like to say? Andy first.
No, but thanks a lot. And we're excited to keep working together on this.
Thank you.
I'm here, ditto. Thanks everyone for joining. This was a great turnout. I'm really inspired by everyone's dedication, curiosity, great questions. So thanks so much for your patience, and we'll see you at the next one.
All right, awesome. Thanks so much. Have a great weekend.